And feel free to chime in if you want to add some content. You should feel free to do that. So I'll just start with this point that the Human Genome Project was a large effort, collaborative effort that took over a decade. And we sequenced the human genome for the first time only in 2003. So this is pretty recent. It's not that old. And we realized that the human genome is approximately 3 billion nucleotides long. Right? So this was quite a seminal moment. We obtained sort of the draft sequence of the genome. And since then, actually, any guesses how much this cost, the first genome, the draft? The total cost of the Human Genome Project. For 3 billion bases, 3 billion letters, how much do you think it cost in total? Just take wild guesses, it's fine. $1 million? A few million? Yeah, actually the answer is about 3 billion. So it took about $1 per nucleotide. You know, it took 10 years and a huge international effort, right? So that was crazy. And now you just fast forward a few years, maybe it's 2020 now, so not 2 decades about. And there's been a revolution in genome sequencing. The technology has really improved throughput. So now you can sequence genomes extremely quickly, you know, usually in a day or so, not for $3 billion, closer to maybe $1,000. The genome sequencing machines have also dramatically reduced in size while increasing throughput. So this is sort of the evolution of the genome sequencers. This is the latest and greatest technology. These are called long-read nanopore sequencers. It looks like a USB device because it is actually a USB device. You plug this into a computer and it directly streams sequencing information into your laptop. We'll be mostly discussing these machines and sequencing information coming out of these second-generation sequencers. I'll sort of give you an overview of how they work today. But since 2003, there's been sort of a super exponential increase in the number of genomes. And the advantage of this is now we don't just have sort of a draft sequence of the human genome, which is like one genome for all of humanity. We're able to sequence individuals and individual genomes, so personalized genomes. And that's allowed us to do what's called population sequencing. And that allows us to identify genetic variation, that is positions at which our DNA sequence varies across individuals, okay, as shown right here. And many of these variations are completely benign. They don't have any, they don't have an effect on disease or something, but they give rise to natural variation amongst us. You can also do interesting case control studies where you sequence or genotype a bunch of individuals. That is, you get their personal genetic variation. And these individuals, let's say, are healthy controls. And you can also sequence individuals with a specific disease. These would be called cases. And then you can perform various kinds of statistical tests to find genetic variants that show strong association with a disease of interest. Okay? These are called genome-wide association studies. This is very commonly used now to map genetic variants, a genetic variation that potentially has a causal association with the disease. Did you have a question? Yeah. Genome-wide association studies. Genome-wide association studies, or GWAS. Okay? So here's an example of what a GWAS looks like. So what you do is for every genetic variant, you can compute a statistical significance of association with a trait or disease. And what you can get is kind of a map that looks like this. So on the x-axis is the genome. On the y-axis is the statistical significance of association. So you can see a negative log 10 of a p-value. Don't worry about how we get this. We'll discuss some of that. Each point in this graph is a genetic variant. It's a position at which individuals have variation in their genome sequence. Okay? And so you can see different dots have different heights telling you how associated they are. And you can see most of the genetic variation in the genome doesn't have a strong association, but certain genetic variants really pop out. Right? Like here's the ApoE locus, a very famous locus associated with Alzheimer's disease, and a bunch of others. So using this technique, you can start very rapidly mapping genetic loci or variants that have an association with the disease. Okay? Now once you do that, so you get all these locations that might be having some functional effect on disease, but the question is, what does it actually do? And so this is where functional genomics comes in. The main idea behind functional genomics is, given a genome, we sort of want to understand what each of these letters and words actually do. So what does this A do in this genome? What does this word do? Does it have any function? Does it not have any function? These are the questions we want to try to answer with functional genomics. Okay? So what you saw before was sort of this is more population genetics and disease genomics, and functional genomics tries to take genomes and tries to understand functionally what each piece of the genome does. So that's just sort of a brief introduction, and now let's jump into some of the internals of DNA and genomes. So I'll give you a quick introduction to genome biology. So just to start off with, what is DNA? It's a long biopolymer. It's made up of 4 basic units. You may have seen them as letters ACGT. DNA stands for deoxyribose nucleic acid. The basic unit is something that looks like this. There's a sugar called deoxyribose, and then there's a base. These are the letters ACGT associated with them. And you can form these long double-stranded molecules, which are basically just polymers of these basic units, okay, ACGT. And DNA forms a right-handed double helix. That means it actually goes this way. Okay, so you'll often see in a lot of advertisements for DNA, they often show you the wrong direction, left-handed helices. General canonical DNA is a right-handed helix, so it kind of goes this way. And the A's pair with the T's and the C's pair with the G's. Okay, and one helical turn of DNA is about 10.5 letters or nucleotides. So the individual letters are called nucleotides or bases, and that's about the length of one helical turn. Okay, and this was, of course, discovered. The structure of DNA was discovered by Watson, Crick, and Franklin in 1953. Here's a YouTube video in case you want to get some information on DNA and structure. Okay. Now the important thing to remember is that DNA is directional. So it actually has a direction. So there's a 5' end and a 3' end, and that's actually related to sort of the structure of the sugar itself. So just to remember, when you're thinking about DNA, it has 2 strands. Each strand has a 5' and a 3' end, and they go in the opposite direction. So here, this strand is going this way from 5' to 3', and the opposite strand is going this way from 5' to 3'. Okay, and this matters a lot because when we download DNA sequences or we analyze them, the directionality will actually have a significant impact on what it actually means. So you can't just reverse a sequence; it doesn't have the same meaning biologically. So just from a computational standpoint, you will often see DNA shown like this. You'll just see one strand of it. Usually it's represented in a 5' to 3' direction. If you want the opposite strand, you need to take the reverse complement. That means you need to flip all the A's to T's, the T's to A's, the C's to G's, and G's to C's, and then you need to reverse the direction. Okay, so the reverse complement of DNA is equivalent to the forward strand. All right, so that's just important to note. When you're analyzing DNA, you will often use the forward and the reverse complement of a sequence as equivalent entities. Yes? Correct. Yes, it is. Yeah, and that has actually different effects on how you actually cleave DNA, mutation rates. All of these things are affected by a lot of these properties. So just remember the chemical polarity. That's very important, and that the forward and the reverse complement are equivalent. And also in the human genome, 3 billion nucleotides are not arranged as one linear molecule. They're actually organized in separate pieces. So inside the nucleus of the cell, you have the genome sort of distributed in 22 pairs of chromosomes. Okay, so as shown here. And you have 22 pairs of what are called autosomes, and then a pair of sex chromosomes, so X, Y. So a canonical male will have X and Y. A canonical female will have 2 X's. Okay, and everybody will have 22 autosomes, 22 pairs of autosomes. Now humans are diploid, so they actually have 2 copies of each chromosome. Okay, so you think of it this way. So each molecule of DNA has 2 strands, the forward and the reverse complement, and then most individuals are diploid and they have 2 copies of each chromosome. So you have 2 copies of each of these, effectively four different strands. Okay, does that make sense? So there's 22 pairs of autosomes, pair of sex chromosomes, XX and XY, canonical female and male. There's also mitochondrial DNA. So there's another organelle in the cell that's called the mitochondria. it is a separate organelle, and it's circular DNA, and it has many, many copies inside a cell. Okay, so the number of mitochondrial DNA copies in a cell are variable across different cell types, and also within a cell you can have multiple copies of the same mitochondrial DNA. Okay, so essentially the diploid human genome has 3 billion base pairs. That's why you call them base pairs, because the bases are paired as double-stranded molecules times 2. You have 2 copies of each if you're diploid. Okay? Yeah, I apologize to the genetics, genomics, biology folks. This is very elementary, but I think for the stats and CS folks, this is a very critical piece when you're thinking about DNA. Okay, so hopefully that's clear. So what does DNA actually do? So there are parts of DNA that have certain important functions. One of these are genes. I'm sure you heard of those. Genes are units of DNA that actually code for proteins. That is, if you read the sequence of a gene, it precisely codes for a particular protein. That is, you can literally translate the sequence, the DNA sequence of a gene into a corresponding protein sequence. And how does it do that? Basically, the DNA is read by certain molecules and then transcribed into what's called messenger RNA, which is another kind of molecule. And that messenger RNA is then translated into proteins. Okay? And there are about 25,000 protein coding genes in the human genome. And surprisingly, the coding parts of the genes, the genome is about 3 billion base pairs, right? But the protein coding parts of the genes are only about 1.5% of the 3 billion base pairs. Okay? So you have 25,000 genes, and the coding pieces of these genes, the ones that can be directly translated into proteins, account for just 1.5% of the 3 billion base pairs. So a very small portion of the genome directly codes for proteins. Okay? And if you ever want to download sort of sequences of genes or proteins corresponding to any organism, these are the 2 most common databases you'll look at. One is GenBank, and the other is Ensemble. And typically you'll see gene sequence represented like this. This is called the FASTA format. So it will have like there'll be a header which will have the name of the gene. In this case, it's some AB000263, and then there'll be a sequence of the gene. And this sequence of the gene is supposed to be from the 5' to 3' direction. Directionality of the gene is very important. The start and end of a gene have very different meanings. And so you have to really keep track of, again, what is a 5' and a 3' end, and on which strand the gene, which strand is being represented in the FASTA file. Okay, does that make sense? All right. So what is messenger RNA? So I said that DNA is actually transcribed by certain molecules into messenger RNA. Messenger RNA is very similar to DNA, except it's a single-stranded molecule. And instead of the deoxyribose sugar, it actually has a different sugar called ribose. And instead of thymine, instead of the T, it has a U. Okay, so it's A, C, G, and U. And if you take a DNA sequence that looks like this, it'll be transcribed into a corresponding messenger RNA where you just replace the A's with U's, the T's with A's, and the C's with G's, and the G's with C's. Okay, so you read the DNA and you convert it into a messenger RNA, which is a single-stranded molecule that looks like this, shown here. The only difference is that there are U's instead of T's that make up the messenger RNA. Okay, and messenger RNA is only transcribed from the coding from genes. All right, not the whole genome. Does that make sense? All right. So these messenger RNAs are then actually converted or translated into proteins. Proteins are also polymers, biopolymers, but instead of 4 nucleotides like RNA and RNA and DNA, proteins are made up of 20 or so amino acids. So canonically 20, there are 2 artificial amino acids as well. They are given these kinds of letters. So this is the table of amino acids. And you can chain these amino acids to generate different kinds of proteins using a peptide bond. Okay, so this is one amino acid, this is another amino acid, and then you can use a peptide bond to actually link them together and create long chains of amino acids. And just like the DNA has a 5' end and a 3' end, similarly proteins have an N terminus and a C terminus. Okay, so that's the directionality of the protein. Proteins can also be represented as FASTA sequences. So you'll often see protein name as shown here with a header, and then there'll be a protein sequence which will be using the 20 amino acids as the basic alphabet. And you can download protein sequences from many databases, one of which is UniProt. Okay, so this is the DNA to RNA to protein, sort of the central dogma of molecular biology. So how is this translation actually done? So there's a explicit mapping from DNA sequence to amino acids. So this is called the genetic code. You have triplets of nucleic acids called codons, as shown here, UUU, UUC, UUA, and UUG. All these four codons, sets of 3 nucleotides, map to specific amino acids. Okay, so these two map to F, which is phenylalanine. These two map to leucine, which is shown right here. And so there's an explicit mapping which is redundant, that is multiple codons can map to the same amino acid. And the genome is actually read exactly this way. You translate DNA into messenger RNA, and then messenger RNA is specifically translated into each of these amino acids based on the codon sequence as you go across the mRNA, okay, as shown right here. So you take triplets and you translate them into specific amino acids, you chain these together and you get a protein or a peptide. Okay, does that make sense for those who are new to this? All right, so the thing is we often think of DNA, RNA, and proteins as these sort of simple sequences, right? But the problem is that actually computationally you can often think of it that way, but they are actually more complex structures. So DNA, for example, does not sit as a linear molecule inside the nucleus. It folds in very complex 3-dimensional shapes, as shown right here. So you have these complex 3-dimensional structures with interactions between distal parts of the genome. So you can have a piece of DNA and another piece of DNA, and the DNA can loop and bring 2 pieces of DNA in contact with each other. And so you have sort of a hierarchical organization of the genome inside the nucleus. And this organization has a very important role in genome function, in how the genome affects cell identity, and also how the genome can be affecting disease. So this is often referred to as 3-dimensional genome architecture. And there's a lot of interesting work going on right now, several interesting assays to measure how DNA folds. Okay? Messenger RNA as well, or RNA in general, even though it's a single-stranded molecule, can fold on itself. So it can form these interesting structures by folding onto itself through base pairing with its own own nucleotides. And these RNA structures actually give a lot of important function to RNAs. So besides simply being a template for translating proteins, RNAs individually also have very important functions. So they will often interact with proteins to create complex molecules, and the structure of the RNA is hence very important. Similarly, proteins, even though they're long chains of amino acids, again, their function is really manifested through the structures that they form. They have these interesting pockets that allow them to bind small molecules like drugs, allow them to bind to other proteins, allow them to recognize specific pieces of DNA. And so protein structure is also very important. And so a really interesting machine learning challenge that has garnered a lot of interest for many decades in this space is simply determining sequence from structure. So if I give you the sequence of DNA, the sequence of an RNA, or the sequence of a protein, can you predict how it would fold to give rise to interesting structures? Okay. The other interesting aspect of these molecules is that they interact with each other. So here's a nice example of protein-DNA interactions. There are lots of proteins that scan DNA looking for specific kinds of sequences and different kinds of affinities for different sequence elements. So the 3-dimensional structure of proteins can recognize 3-dimensional structure of DNA and bind them in very specific ways to manipulate how DNA is accessible. In fact, these are exactly the kinds of molecules that transcribe genes. So they have very important roles in gene regulation, as we'll discuss. Similarly, proteins can interact with other proteins to form complex molecular machines. And so there are these protein-protein interaction networks, very complex networks of how proteins interact with other proteins. And similarly, also proteins, as I said, have strong interactions with small molecules. This is actually how drugs work. They are small molecules that have specific bind in specific pockets of proteins, and they have certain effects on the conformation of the protein and the properties of the proteins. So for all these different aspects of drug discovery, chemical informatics, and so forth, interactions are a major aspect. And so a lot of machine learning methods have been developed to actually predict interactions, understand interactions, try to figure out what features in the DNA or the protein result in specific interactions, and then finally how to engineer DNA or proteins or small molecules that will give rise to specific interactions. Okay, so that's another interesting machine learning challenge. So these are, the reason I'm mentioning this is that all of these aspects could be interesting topics for you to focus your projects on. Okay, and we'll be going through several aspects of them in the course as well. So how is DNA transcribed to RNA? What you have is, this is the basic structure of a gene. You have a start end of the gene and the end stop end of a gene, and you have this molecule called RNA polymerase that binds upstream of the start site, the 5' end. This is often referred to as the core promoter of the gene. The promoter of the gene is the place where the RNA polymerase binds. It's a protein complex. What this does is open up the two strands of DNA. It'll read one template strand and then create a corresponding messenger RNA out of it by simply transcribing, as I said before, just the A to U and G to C mapping. That's how you create messenger RNA. And this messenger RNA then is in the nucleus. It is transported out of the nucleus and then translated into proteins. So there are interesting machine learning problems associated with just this process. Things like predict all the genes in the genome. Can you, just looking at DNA sequence, can you predict where the genes are? Where are the start and end sites? So predictions of what in the DNA sequence or determines the start site and the end site of a gene. There are more complications beyond this. For example, the messenger RNA that's just produced from transcribing DNA actually is then processed in many interesting ways. So genes, when they're transcribed, have different pieces in them. Some of these pieces are called exons, and the exons explicitly code for amino acids. And there are other pieces of the gene referred to as introns, and those don't actually those don't directly map to amino acids. So genes can be quite long. So here's an example of a gene. This gene is 200,000 nucleotides long. But it codes, most of the gene actually does not directly code for proteins. So it has 26 exons, as you can see here, these little red bars. These are the exons, and they are directly translated into protein. But these intermediate pieces called introns are not used to translate to protein, but they're still part of the gene. So the whole 200,000 nucleotides is transcribed. And then there's a process called splicing, which actually removes these introns and then fuses the exons together. Okay, so splice machinery comes and takes the pre-messenger RNA, which is the raw messenger RNA that pops out after you transcribe the DNA, and then it slices these introns out and then fuses these exons to create mature messenger RNA that is then translated into protein. Now, why is this important or relevant? Well, you can create interesting combinatorics. So if a gene has 26 exons, you may not necessarily have to splice all 26 together, one after the other. You can selectively splice different combinations. So I can, for example, exclude exon 2, right, and splice exon 1 and 3 together, and 4, 5, 6, 7. So by having this exon-intron structure, the cell can use combinatorics to create many more proteins than what one gene can essentially encode for many proteins by combinatorial splicing of exons and introns. Does that make sense? So I can take 10 exons and I can combine them in many different ways to give rise to a whole number of different kinds of mature messenger RNA transcripts or isoforms, and each of those isoforms will give rise to a different protein. So this is how 25,000 genes in the genome can actually give rise to a much larger diversity of peptides and proteins by combinatorial splicing of alternative splicing of different exons with each other. Yes? Yeah. So the sequential order is usually preserved, but you can skip exons. You can combinatorially skip them. And there are specific signals again in the sequence that drive a lot of this. So there's a lot of overloading of the sequence, right? So you have the codons in the exon that code for the corresponding amino acids, but exons also contain subtle signals that determine where these splice proteins bind and how they splice in which conditions. Okay, so there's also interesting information encoded in the DNA sequence. So another interesting machine learning challenge is, can we predict all these splice sites, gene isoforms, and alternative splicing events? Okay. So then what is gene regulation? As we saw before, DNA sort of is transcribed into RNA, and that is translated into protein. Gene regulation really determines when, where, and how much of this happens. Okay? So it's the control system of the genome determining when and how much of messenger RNA should be produced and which cell types, and how much of that should be translated into protein and so forth. So that's really where gene regulation kicks in. So to understand gene regulation, and we have to think about sort of what is the control system of the genome, and for that we'll sort of just open up the genome and try to study its anatomy in a bit more detail. So here's the cell, here's the nucleus of the cell. So we've taken the chromatin, the DNA fiber. I'll explain what these things are, and this is chromatin. But let's just look at the DNA for now. So here's the DNA, we open it up, and we talked about there being genes, right? So these are two genes right here. This gene is an active gene, so it's producing proteins or messenger RNA, and this is a repressed gene, that is, it's been turned off for now. So how are genes turned on and off, right? So as I said before, only 1.5% of the genome directly codes for proteins. Okay, so 99% of the genome is what's called non-coding. It's not directly being read out to translate into protein. So what's interesting is the remaining 99% of the genome is sparsely populated with a whole bunch of sequence elements whose role is regulatory. Their role is to control activity of genes. Okay? And so again, there are little pieces of DNA as shown right here. We talked about one such piece, which is the promoter of the gene, which is always just 5' upstream of the gene itself. That piece of DNA has various sequence elements that are attractors for specific proteins. Okay? And those proteins come together and they assemble the RNA polymerase complex that then transcribes the gene. Now, in the human genome and in many mammalian genomes, you not only have these control elements, regulatory elements or pieces of DNA, just upstream of the genes, but also in other intergenic regions, just scattered around all over the place. So these little orange things, they need not be right next to the gene. They can be anywhere in the intergenic space. There are also a bunch of these in introns of genes. Okay? And what they do is they act as binding sites for various proteins. So the sequences in these regulatory elements, as I said before, they're recognized by specific proteins. These proteins are regulatory proteins. So they recognize specific DNA sequences based on the structure and sequence. They bind there. And what they do is once they bind there, the DNA will often loop. So this piece of DNA will loop over to this gene and bring the protein complex that's bound here to the protein complex that's bound here. And collectively, they will recruit RNA polymerase, which will then transcribe the gene. And this is a very dynamic process. So this encoding in the non-coding genome as well, while the coding part of the genome codes for proteins, these non-coding elements, regulatory elements, their sequences also have a code. But that code is recognized by regulatory proteins that then activate or repress genes. And what's interesting is these regulatory elements and various other parts of the genome are also flagged by various biochemical modifications, as shown right here. So these red, orange, yellow, green, gray modifications, this is real data, by the way. I'm making it look like a cartoon, but these are actual modifications. You can see how this gene is active, and its gene body is marked by this green biochemical modification. And it perfectly delimits the gene, right? You see right here. Also, the promoter of this gene, this active gene, is marked by this red and orange marker. These other regulatory elements, these distal ones, often called enhancers, are marked by this orange or yellow marker. And this whole region of the genome that is repressed does not have any of these markers, but has this gray marker. Right? So the cell uses various biochemical modifications to tag pieces of DNA or the proteins that the DNA is bound around, almost like molecular memory. It tells it like what piece of DNA is performing what function. And so even though the DNA sequence is static, by being able to add and remove chemical modifications to the DNA, we can effectively, the cell can add dynamics to the static DNA piece of sequence, right? Proteins can bind a piece of DNA or not bind it. You can add or remove a molecular flag. And by adding or removing these molecular flags, these various kinds of molecular events that interact with DNA, we can have the genome express itself dynamically, respond to the environment, be different in different cell types, and so forth. So to understand what this is, we have to go a little bit deeper. These cylinders that the DNA is wrapped around, they have very important functions, okay? And those are referred to as nucleosomes. So yes?
Yeah, what percent of the DNA play a regulatory role?
That's a great question. I cannot give you an exact answer because that's still very debatable. We definitely know 1.5% of the genome is coding. The estimates for how much of the genome is actually regulatory ranges from 5% to 25%. Okay, so the reason is because you can use all of these experiments to measure these modifications and come up with hypotheses. You can predict which elements might be regulatory. The issue is that there are so many of them that they have not been all validated. So the predictions range hence from 5% to 20%, depending on how you define a regulatory element. And we'll get into some of that as we go forward. But it's very fascinating. There's an entire piece of the genome we don't understand. So even though we know kind of where all the genes are, we don't know what most of their functions are. And we know very little about the control system of the genome. And this actually has a very important role in disease and natural variation across individuals as well. So going a little deeper, the DNA is not sitting again naked in the nucleus. It's a very long molecule. It needs to be stabilized. So it's structurally wrapped around these cylinder-like things. These are called nucleosomes, okay? And the nucleosomes, as shown here, are made up of basic proteins known as histones. And there are 4 types of histones, H1, H2A, H2B, H4, and H3, okay? So there are 8 of these that make up one histone, one cylinder. And remember, these are proteins, right? So they are chains of amino acids. And the tails of these amino acids can be modified with all of these different modifications. You can add an acetyl group, you can add a methyl group, you can add a ubiquitin group. So all of these modifications can be made to every residue in the tail of each of these histone proteins on any of these nucleosomes. So you can imagine the complexity of this kind of epigenetic code or the chromatin code, right? Because I can add and remove all these modifications to any specific histone on any specific nucleosome in any specific cell. So these modifications, often referred to as chromatin modifications or histone modifications, you'll often see them written like this. There'll be something like that looks like H3K9Me3. So the way to read this is histone 3, K is the residue, lysine, K is the amino acid, right, which K stands for lysine amino acid. 9 is the position, so that's the 9th lysine in the tail. And you add 3 methylations. That's how you read this code. Okay? So H3K9 trimethylation means we added 3 methyl groups to the 9th lysine on histone 3. Then you have another modification here, which is H3K27 Me3, which is 3 methylations to lysine 27 on histone 3. So there are many, many such modifications, thousands of these, and they can be combinatorially added or removed on specific nucleosomes, right? So that's how you actually add this molecular information, which allows you to take chromatin and add this sort of epigenomic information to it. Also, DNA itself can be directly methylated. So the C's, ACGT, right? The C's in DNA can be actually methylated, and you can have methylated C's. That's another kind of epigenetic modification. So in this case, you're not modifying the sequence, right? The sequence is the same. You're simply adding or remove methyl groups to the C's. That's DNA methylation. Or you can add and remove all of these molecular flags to these tails of these histones that make up these nucleosomes. And these nucleosomes are all over the DNA. So the DNA is wrapped quite regularly around these nucleosomes. Okay? So the millions and millions of these nucleosomes all around the DNA, they can be popped on and off, and you can modify them. And these proteins that need to bind DNA, they also compete with these nucleosomes to get access. So the nucleosomes also have a regulatory role because they act as competitors to these other proteins that bind DNA to regulate gene activity. Does that make sense? Okay. So that's sort of a very basic anatomy of the genome, sort of structural aspects of the genome and a very high level view, but sort of all of these aspects will be very important because a lot of the data that we'll be talking about in the next few lectures and the machine learning methods will be applied to these kinds of data sets, measuring all of these different kinds of modifications and so forth. Now the other interesting aspect is that humans are multicellular, right? Multicellular organisms. There's a hierarchy of tissues as shown here. There's all these different organs. tissues. Within the tissues, you have different cell types. And all of these cell types largely contain the same DNA in a particular individual. So how does this one genome sequence give rise to all this diversity of function in the human body, right? And so the trick really is to simply dynamically use the genome and express it differently in different contexts. So if I take a particular gene in the genome, this is CREB1, and I look at its gene expression in different tissues, so each row here corresponds to a different, each column here, each tick here is a different tissue. You can see different parts of the brain. This is adipose, there's esophagus, there's colon, and so forth. You see there's lots of variation, right? So the same gene is expressed differently in different cell types. So I can make different amounts of proteins and genes in different cell types and thereby give rise to different sort of cell states and cell types and function from the same genome, right? And that's why gene regulation is very important, because that really determines cell identity and cell function. Which proteins are produced and how much amount at what time determines cell identity and cell state. This gets more complicated because so far we are talking about like anatomy, tissues, you've got brain, you've got liver, you've got kidney, but you've got much more complexity because within each tissue, you've got a whole variety of cell types, okay? And so what's really interesting now is this has really happened just in the last 3 or 4 years, is we have this ability to now measure all of these activity levels like gene activity, gene expression levels in individual cells. So far we used to be able to do it in bulk populations. So you take the liver, we just crush it up, and then we take all the cells, all the RNA or the DNA and measure it, right? So we'd get sort of one measurement. This is an example of what it would look like. You take the tissue and you get one measurement across all the cells in the tissue, sort of an aggregate average view. And so each of these represent different genes. So you say, oh, look, this black gene is expressed this much, and this red gene is only expressed this much, right? So you get a quantitative value of how much each gene is expressed in each tissue. But actually, if you look at the individual cells, you see all this heterogeneity. You've got these 4 types of cells, and in this yellow cell, this red gene is expressed quite a lot, and in these cells is not expressed at all. And you're just mashing this up and getting these average tissue level expressions, right? Now you can start measuring all of these things at the single cell level. And so amazing new set of machine learning problems has popped up. We don't even know what cell type and cell state is anymore. Like earlier we used to say, oh, there's a liver cell, and there's a kidney cell, or whatever, there's a brain cell, there's a neuron. Now we're realizing there are 50 different types of neurons, right? Each of them in different states. Also, when you treat a neuron with a certain kind of stimulant, it goes into a different state. Some of these states are very transient. They only found when one cell type, transitions into another cell state. For example, immune cells, right? T cells and B cells, when they come into contact with different kinds of stimulants, they will go through various different kinds of cell state transitions. And it was very difficult to measure these before, but now since we can do single cell measurements, there's all these interesting questions of unsupervised learning. If you give measurements of gene expression in individual cells, thousands of them, you get this kind of matrix. You get genes by cells with values, and your goal is to now start figuring out what's the heterogeneity. Can I figure out subpopulations of cells that behave similarly, and are those subpopulations different from other subpopulations? And so now there's a whole quest. There's this thing called the Human Cell Atlas, which is an international consortium. Everybody working together to try to understand what does cell type mean? What does cell identity mean? What are the different types of cell states and so forth? And so we'll discuss a lot of these methods as well towards the last part of the class. So this is gene expression, but then the big question is, if genes are being expressed differently in different cell types, they must be regulated differently by different elements. So that makes us go back to the non-coding genome, right? Because we're looking now at, so this is an example of some data from one of the consortia. Each row here corresponds to a different cell type. In this case, they're tissues or cells, or sort of bulk tissues or bulk cell types. What I'm showing you here is a piece of the genome. Here's this gene called PAX5. And what we're going to see is the same piece of the genome, how it is dynamically activated or repressed in different cell types and tissues. Okay, so the rows correspond to different cell types and tissues, and the colors corresponding to these different types of elements and different activity states. And the way we've inferred these colors is by looking at these chemical modifications. These combinations of modifications, these red, orange, yellow flags which you can measure. From that we can sort of infer the state of any piece of DNA in any single cell type. Okay? We'll talk about this in more detail. It uses machine learning methods to figure out these states. But after we do this over hundreds and thousands of different cell types and tissues, we start getting an estimate of how many control elements there might be in the genome. So there are about 25,000 protein coding genes, and to regulate these genes, this is where the estimate kicks in. The estimate is about 2 million control elements are likely there. They show signs of being control elements. They have marked by these various modifications, and many of these are utilized in a very cell type specific way. Just to give you an estimate of how this looks like, I'm taking the same piece of the genome and I'm showing you how the modifications or the sort of the state of that piece of DNA is differentially being modified in different cell types. So these are all embryonic stem cells, okay, each of these rows. So you can see this piece of the genome is all gray. Gray means repressed, okay. Now if you look at these are different kinds of blood cells, immune cells. You'll notice that the same piece of the genome is again gray in many of them, but in this specific cell type, notice what happens. The 5' end, the start of this gene goes from being gray or pink to red, so it was activated. And the gene body goes from being gray to green, right? The specific coordination of the promoter turning on with a new kind of modification and the gene firing with a new kind of modification. And if you look at all these other types of tissues and cell types, these are epithelial cells, these are brain tissues, these are muscle, heart, smooth muscle, digestive. This gene is off in all of them. It's a very cell type specific gene, right? It activates only in this particular type of immune cell. And if you look at all these other genes nearby, you see all this interesting variation, different genes being activated in different cell types and tissues. And if you look at all these yellow and orange elements, you'll see also them turning on and off, lighting on and off, right? So this is beautiful dynamic sort of regulatory elements being marked by different biochemical markers in different cell types and tissues, activating different genes and activating and repressing them. And so this is sort of the dynamic language of the genome, the same piece of DNA. The sequence is the same in all these cell types, but they're dynamically being activated, repressed by different proteins, by different modifications, and so forth. So there are interesting machine learning challenges here. Predict regulatory elements and their cell type specific activity. Which regulatory elements regulate which genes and which cell types, and can you predict gene expression from regulatory element activity, right? So this is sort of trying to predict the control circuitry of the genome. If you figure this out, you can then start manipulating the genome. You can understand how mutations in specific elements might affect gene expression and cell state and so forth. So just to focus again on the dynamics, if we take all of these yellow and red elements and we count how many there are, as I said, we find about 2 to 3 million of these. They're pretty short. They are anywhere ranging from 100 to 200 nucleotides in length, okay? And so that's, in this case, we have 2 million of these columns, and these are the hundreds of different types of cell types and tissues, and we're simply coloring them by whether they are active or not in each of those cell types. So if we organize these using a clustering method, you start seeing these really beautiful modules of elements which are specifically active in different lineages, right? So here you have a specific set of elements that are active only in ES cells. These are specific to T cells and so forth. And so the big question is, what is in this sequence, in this collection of sequences that makes them active only in one specific cell type? There must be something in the code. There must be some sequence in the code in the sequence that makes them specifically active in that cell type. And so if you take these pieces of DNA, these elements that are co-activated in a specific cell type or lineage, and you look for little words, right? You start seeing that there are certain words that are overrepresented. You see the same words sort of repeating themselves in those elements. And what these words happen to be, these are often referred to as sequence motifs. They happen to be these kind of sequences that are recognized by specific regulatory proteins called transcription factors. Okay, so these are the regulatory proteins that look for these specific sequences, and they preferentially bind to those sequences. So if you take a piece of DNA and you have this sequence in it, this protein will preferentially bind to that piece of DNA. So the genome essentially has evolved to have very specific sequences or little motifs or combinations of motifs in different elements, and those kinds of elements will co-activate in specific cell types because different proteins, specific proteins will bind those elements and then recruit RNA polymerase to regulate nearby genes. So really interesting machine learning problem is to try to decode the language of these non-coding elements. What is the code? Just like there's a genetic code, there is a regulatory code, and it's much more subtle, much more combinatorial. It's not this simple table of codons mapping to amino acids. You need to try to figure out what in the sequence is driving all of these effects. So these are sort of the multiple facets of the regulatory code. You have any piece of DNA that is acting as a regulatory element typically has these nucleosomes. These nucleosomes have histones. These histones are modified with these different chemical modifications. The regulatory proteins that bind the DNA encoded in this regulatory element, they recognize specific sequence motifs. There's a combinatorial code of little words that allow different combinations of proteins to bind. And also remember, these proteins have to compete with these nucleosomes. There's interesting spacing orientation constraints on where proteins can bind, how they compete with nucleosomes, how that results in chemical modifications being added or removed. Okay? So all of these are interesting machine learning challenges. So moving one step forward, we mostly talked about, yes?
On the last graph with the sequence of like the relative size of the letter represents like the frequency compared to the other letters. What is like the overall type on the graph?
Yeah, we'll discuss that a lot more in detail going forward. But yeah, the key point you've noticed is these are not deterministic words. So there's a fuzzy preference to these words. For example, what this means is at this position, the protein binds to motifs that always have a G. Okay, whereas at this position, you could have a C or a G. So the height of this letter is actually the information content if you take a collection of these motifs and you collapse them. And so there's a probabilistic interpretation to these motifs. So it's an affinity landscape. You have, if you take k-mers, short words of size, say, 12, the protein will have sort of a whole landscape of affinity to all of these different words, and you can collapse them into a representation that looks like this. We'll talk a lot more about this in a few lectures down the line. So far we've talked about the internals of the genome and the control circuitry, and we talked about variation across cell types and tissues. But we also talked about variation right in the first slide. We talked about variation across individuals in our basic DNA, right? So we said that if we take a bunch of individuals and we sequence our genomes, we'll find that we differ at specific positions. And this is referred to as genetic variation. So as I said, each of us is diploid. So we typically have 2 copies of each chromosome. And so at each position, we could have either the same letter or we could have 2 different versions of it. So if we have the same letter in both our copies, that particular variant is supposed to be, each of the 2 versions are referred to as alleles. Okay, those are the 2 versions of each letter. And if you have the same alleles at a particular position, you're referred to as being homozygous. So you have identical alleles. Some individuals may be heterozygous, that is, they have 2 different copies, 2 different letters at the same position. Okay, and basically individuals will vary across at this position across populations and across individuals. So one of these alleles is often referred to as the major allele, and the other allele is referred to as the minor allele. The major allele is the one that's more frequent in the population. So if you take a bunch of individuals and you see how often you see A versus C at this position, maybe you see the A more frequently. That would be referred to as the major allele, and the less frequent one would be referred to as the minor allele. So you can often refer to genetic variants by their minor allele frequency, which tells you the frequency of the minor allele. Okay, tells you how frequent there's a variation of that position of the less frequent allele. There are other kinds of variations. So these are single nucleotide variations, right? So you're just changing, these are like mismatches. At particular positions, you're changing the letter. But you can have more complex ones. You can have these structural variants. For example, you can have insertions, you can have deletions. So let's say this is the reference genome, and you have 1, 2, 3 pieces of DNA, you could insert. Some person could have an insertion. That's a bunch of nucleotides that just get inserted in there. You could have a deletion, so this 3 went away. You can have an inversion where this piece of DNA came this way. You can have copy number variation where you have multiple copies of the same piece of DNA replicated. You can have tandem duplication. So there are a variety of different complex kinds of variation beyond just simple mismatches. Now for these single nucleotide variants, based on the minor allele frequency, they're often classified as common or rare. So common variants are those that have typically a minor allele frequency of greater than 2 or 5% in a population. Okay, 2 to 5%. So if you're more than that, if that variant has a minor allele frequency more than 2% or 5%, it will often be referred to as a common variant. If it's less than 2%, then it's a rare variant. And if it's present only in a single individual, you'll refer to that as a private variant or a mutation. Yes?
When you're defining these SNPs, are they defined like comparing specifically like the same tissue type across those individuals, or is it, or like can you define a SNP from like a full tissue type?
Usually it's just done from blood samples because the key point is that the assumption, which is not entirely true, because there's mosaicism actually even inside an individual. That is, you can have genetic differences between cells inside a single individual, but on average, that variation is much less than what you see across individuals. So if you just take DNA from any tissue, it will have on average the same sequence. So it will be a good representative of your genome. So you just take, usually this is just taken from blood because it's the easiest you have access to. Oh no, these can have different, there can be different kinds of variation, so we'll get to that. This is just your genome, right? And you have the human reference and then you have variants, and they can be single nucleotide variants or they can be these copy number variants. All of them will have allele frequencies, and then you have common and rare and private, okay? So here's the question that you had, is where do these variants lie, right? Are they in genes or are they outside genes and so forth? So based on where they lie and what they do, coding variation can be of different types. So you have what's called synonymous, non-synonymous, nonsense, and frame shift variation. Okay, these are all different types of coding variation, that is genetic variants that fall within protein coding sequence. Okay? So this is an example of a synonymous variant. Here's the sequence, okay, and this A is converted to a G. Now, this is the codon, UUA. It gets converted into UUG, right, because you have that variant. But based on a genetic code that maps to the exact same amino acid. So the protein that's produced remains the same. And that's referred to as a synonymous variant. A non-synonymous or missense variant will actually change the amino acid. Okay, so you potentially have a different protein. At least the sequence of the protein would be different. You can have a nonsense, a mutation, as shown right here, which will create what's called a stop codon. The stop codon basically says stop translating. So it will truncate the protein. Okay, so this is obviously very bad. If you have a variant that produces a stop codon, somewhere in the middle of a protein sequence is going to create a truncated protein, which is obviously not going to function properly, right? And then you can have frame shifts, which, for example, here you have like a deletion, and that results in a complete shift of the codon sequence itself, because you're reading them in frames of 3, right? If I remove one of the nucleotides, all of a sudden everything shifts, and that produces a different protein as well. So these are different kinds of coding genetic variants, okay, based on what effect they have on the property of the protein. You also have a lot of non-coding genetic variants. They can affect splicing or regulatory DNA, so they can affect these motif boards, right? Maybe at one place I need to have a G. If I make that G into an A, the protein can't bind anymore, right? So you can have a big effect on sort of how proteins bind and how they affect regulation. There's a lot of non-coding genetic variation as well. Many of it is non-functional, some of it is functional. Yes?
So out of these three errors, is there one that's like particularly
Yes.
dangerous or toxic for the organism?
So the stop codon would be the worst. The synonymous one would be the most likely benign. Like it could change, for example, the rate of translation. That has often been seen. But it will at least not change the protein sequence itself. So the structure of the protein will be intact, everything will be fine. So synonymous usually are considered benign. Missense can be pretty bad, depending on how important that amino acid is, what its function is. Stop codons and frame shifts are horrible, right? Because they will entirely change the sequence. I mean, it'll either truncate the protein or it'll produce the remaining part of the protein will be completely different because it will be translated differently, right? So those two are usually the worst. So this is based on sort of coding variation and the non-coding variation. This is based on a lot of the functional effect of the variant. And then you have two other kinds of variation depending on the type of cell in which the variant arises. So you have germline mutations. Germline mutations happen in the sperm or the egg, and these get transmitted to the next generation. Or you can have your genome, and then which you inherited from your parents, right? And then somehow, as you were growing older or whatever, you ended up with a mutation in some random cell type, like in your liver or in your brain or whatever. And these are referred to as somatic mutations, okay, because they are in somatic tissues, they're not in germlines. These will not be transmitted to the next generation because they're not in your germline, okay? Somatic mutations are very common in cancer. The cancer has a lot of somatic mutations. They have germline variants as well that can predispose you to cancer, but they're often driven by specific mutations in specific tissues that then give rise to tumorigenic behavior. So this, genetic variation is very important because now once you sequence a person's genome, we'll get several thousands or millions of variants, right? And now the question is, which of those variants are actually good, bad for different kinds of diseases, traits, environmental contexts, and so forth. And so now there's a whole amount of work going on in trying to predict genetic variants from genome sequencing data. So if you're sequencing someone's genome, how can you accurately provide them genetic variation? That's clinical sequencing. Which genetic variants are benign versus harmful? That is, which genetic variants are associated with just natural variation of traits like hair color, eye color, skin, whatever. And which of those actually might be associated with disease, right? What is the genetic architecture of disease? So if you sequenced a diseased patient's genome, which of the variants in combination are predisposing them? Which ones are the causal ones? Which ones are not directly affecting? Similarly, in cancer genomes, which variant somatic mutations are driver mutations, which are passenger mutations? These are important questions. And most importantly now, even if we figure out a causal mutation, if you want to do something about it, we need to figure out the mechanism through which it's acting, right? So this is where we link genetics or population sequencing to functional genomics, all of this other stuff we talked about. That is, how does a genetic variant affect protein coding sequence or regulatory properties of DNA or gene expression? All of these become important questions. Okay, so that's sort of a very high-level view of all of biology in sort of 40 minutes. We'll be diving much deeper into each of these aspects as we go forward. I just want to introduce you to sort of the main areas, challenges, and where ML and statistical methods play an important